So now looking at the patterns of inheritance, specifically patterns of inheritance, what we're supposed to be looking at really is using our monohybrid and dihybrid crosses to solve problems. We're going to be looking at how the information we gain from the monohybrid and dihybrid crosses can be useful under different circumstances, right? And then in order to use these things, we're going to be defining terms like dominance, codominance, incomplete dominance, epistasis, sex linkage. We're going to be speaking about type of stuff like that, right? And then we're going to be moving into the chi-square test, right? And how we apply genetic crosses, right? How we analyze the results of these genetic crosses, these dihybrid crosses and monohybrid crosses by applying the chi-square test, right? We're going to be looking at the different hypotheses that we get from that, and then we're going to basically just understand the importance of the ratios of the observed and expected traits that we're looking at, right? And I have a scenario that we're going to be looking at in order to work through what each piece of chi-square test really speaks about, right? So let's begin with the genetic terminology, right? So with every genetic section, we're going to be talking about genetic terminology, and we first start here with genes. So we know that the genes is the basic unit of heredity, right, within DNA specifically, right? So it consists of a sequence of DNA which codes for a specific protein or functional RNA molecule, and that is the central dogma of DNA replication. We're going to be speaking about that a little later, right? So genes are fundamental units of inheritance, transmitting traits from parents to offspring, right? So I have examples for all of these. So let's say that we're studying eye color in humans, right? And we look at a specific gene. We have a specific gene that is responsible for eye color, which is located at a specific region, a loci of a chromosome, right? So there are different variations of the genes that we call alleles, right? And that's what we're going to be looking at here. So an allele is one of the alternative forms of a gene that can occupy a specific location or position or locus, right? Or loci on the different chromosomes, right? So alleles can result in different expressions of a trait. So let's say that we have the gene for eye color, but one allele might give us blue eyes, one allele might give us brown eyes, one allele may give us chestnut eyes, right? Different types of alleles in combinations in more complex situations may give us the situation of the chimeric eye, right? We have different types, right, of alleles that look at different stuff. So each parent contributes one allele. Okay? Everything's great so far, right? These are terms that you guys understand, right? Okay, lovely. Right? Dominance. A dominant allele is one that is expressed phenotypically in heterozygous, or in heterozygotes, right? Masking the effect of the recessive allele. When we talk about phenotypically, what is phenotype and what is genotype? Could somebody make the difference? What is genotype and what is the phenotype?
Phenotype are like the physical features that you can see, and the genotypes are the features that are on the gene, basically.
So what we use to represent the genotypes, lovely, thank you for that response. Right? Well, the genotype we're looking at, right? That is really exemplified by letters. We use letters, right, to really look at that, right? And then the phenotype is what is seen. Which affects which though? It is the genotype that affects the phenotype, right? Lovely. So it's the different types of alleles that affects how the organism presents, right? And you know that that is not the only thing that affects phenotypes. The environment can change the phenotype of an organism as well, right? Nutrition can change the phenotype of an organism itself. Because the organism may be naturally tall, but because they live in a nutrient deficient environment, they are short, right? So based on nutrients, based on what the organism consumes, the environment can affect how they present. right? Let's say in the case of contemporary Caribbean society, in which we have, I'm not going to mention my staunch views on this, but I highly it's completely distasteful, right? In the case of bleaching, right, where persons are born with specific amount of melanocytes, right, that gives them their pigment, right, their genotype is saying that you are going to be expressing a specific phenotype of this shade of skin tone, right? But you can change that shade using different types of chemicals. The environment and other things that you do can change the phenotype of an organism. All right, but we're not going to be talking about that just yet. We're going to be looking at the genotype and how it directly affects the phenotype, not any other effects. Okay, so in dominance, we know that one allele is expressed phenotypically in heterozygotes, right, over the other. So if a brown eye color is dominant over blue, right, a person with one brown allele and one blue allele, which is heterozygous, right, would have brown eyes, as the dominant allele is brown, and that is expressed. Lovely. Lovely. In recessive alleles, now recessive alleles is one that is only expressed phenotypically in homozygotes when the dominant allele is absent. So we're talking about homozygous recessive. So in recessive homozygotes, we're going to have that recessive allele being displayed. So yes, in the case that blue eyes are recessive and brown eyes are dominant, if we have two copies of the blue allele, if we're homozygous recessive in that case, then the dominant allele itself would not actually be able to mask, right? And the blue allele would actually be presented, right? Because we have two of the blue alleles and absence of the brown allele. Codominance. Codominance occurs when both alleles in the heterozygote are fully expressed, resulting in the phenotype, right, that shows traits of both alleles. For example, in the AB blood type, right, that's an example of codominance. If an individual inherits an A allele from one parent and the B allele of another, both A and B antigens are expressed on the red blood cells. Therefore, we have the creation of an AB blood type because both are expressed equally. This is codominance. All right? That is what's happening there. If there are any questions, remember to stop me because I'll answer any questions. All right? That's codominance there. Incomplete dominance. Incomplete dominance occurs when the phenotype of the zygote is an intermediate between the two, right? Between the phenotype of the zygotes, the homozygous dominant and the homozygous recessive individuals, right? So we're gonna have incomplete dominance. So for example, in the snapdragon flowers, which I put on the screen there, right, a cross between a red flowered plant, right, and a white flowered plant produces pink flowers, illustrating incomplete dominance. So codominance is when both are shown, incomplete dominance is when they mix. All right. Homozygous, what does that mean? Homozygous refers to an individual having two identical alleles at a specific gene locus on homologous chromosomes, right? So that's the definition. So if an individual inherits two brown alleles for an eye color, then they will be homozygous for brown eyes, right? Well, let's say that they're heterozygous. Heterozygotes now, right, tend to speak to individuals having two different alleles at a specific gene locus on homologous chromosomes. Go ahead. You raise your hand.
So sir, with incomplete dominance, both the alleles are homozygous, but they just mix.
In that case, yes. So if we have two dominant alleles, right, and then we mix like that, we're gonna have that. So are they homozygous?
So they have to be dominant.
They're gonna be dominant, yeah.
Okay, sir.
Alright, they have to be dominant in some way with each other. Alright. For heterozygous, no, we're basically speaking about the individual having two different alleles at specific gene locus, right, on homozygous. not homozygous, homologous chromosomes, right? So even an individual inherits one brown allele and one blue allele, right? Then obviously the heterozygote color will appear with accordance to dominance and recessive, and whether or not the allele is dominant or recessive, right? So you're gonna have two different ones, right? And most likely in heterozygotes, right, the dominant one is really shown. All right, if there are two dominant, then it's going to be either co-dominance, right, or we're going to have what we call incomplete dominance. All right, sex linkage, right? So sex linkage refers to the inheritance patterns of genes located on the sex chromosomes, right? So these are the chromosomes that we see in our gametes, leading to certain traits being more common in one sex than the other. For example, hemophilia is an example of a sex-linked disorder where the gene for blood clotting factors is located on the X chromosome. Since males have only one X chromosome, they are more likely to express hemophilia if they inherit a defective allele. So males tend to be more hemophilic. All right, and we tend to most likely get them from their mothers. The color blindness as well. Right, so this speaks to when, right, the gene itself for a specific trait is located specifically on whether on a X chromosome or a Y chromosome, right? So if it is Y chromosomal, we know that women won't show it, right? But men would show it because men are XY and women are XX, right? All right. So multiple alleles, no. So multiple alleles refer to the presence of more than two alleles for particular genes within a population. So there are more than two alleles for particular genes. So the ABO blood group system is an example of multiple alleles where there are three alleles, right? A, B, and then the smaller I, right? So it's really anti-A, anti-B, and then we have just I, right? That determine the blood type in humans. So blood type is an example of what we call multiple alleles. We have multiple alleles for the expression of the genes, right? And here we have it presented here. All right. So we have group A and group B. When group A and group B combine, they give us co-dominance. And there is a case where we just have none. So that's group O. All right. Epistasis or epistasis. Epistasis occurs when the expression of one gene masks or modifies the expression of another. So it is not dominance. It's similar, but not dominance. It's just not dominance, right? For example, in the coat of Labrador retrievers that I have on the screen here, the gene for pigment disposition B determines whether pigment will be deposited in the hair, while another gene E determines whether the pigment will be expressed as black or yellow. The expression of the gene E masks the expression of the gene B, illustrating epistasis. Same thing for albinism, right? In albinism, we have a gene that tells us how much melanin will be produced, but there's another gene that will tell us whether or not melanin will be produced at all. So that is epistasis or epistasis, right? So that is what we have there, epistasis. Okay, so those are all the genetic terminology that we shall be using for the rest of the topic. All right. So if there's a piece of genetic terminology that you don't get, let me know. All right. Are there any questions or concerns? Go ahead.
Sir?
Go ahead.
Yes, sir. You can go back to epistasis again. It really similar to dominance. I mean, I see a difference.
Okay. So, let's see if we can. All right. So,
Sir?
Yeah, so the epistatic gene
You can hear me,
specifically.
sir?
Oh yes, I can. Let me see.
Can you hear me? I was asking if you could go back over epistasis because I'm not seeing the difference between it and codominance.
All right. So with the epistatic gene, right, we have what we call an E gene, right? So if we have an E gene, we'll just write the E gene there. Right, we have an E gene, right? Oh, that's not an E, right? Let's say that we have if we have an allele B, right? Large B, and we have allele small b, right? So large B gives us a black coat. We just put BLK here. And a small b gives us brown. ERN. Right? Following so far? So we have two alleles that give these two, right? Now, what we're having now is demonstrating epistasis, right? Epistasis determines whether or not the genes will be expressed at all, right? So what epistasis can modify the expression of the gene, right? So it can actually inhibit it. For example, let's say that you are actually heterozygous in this case, right? And you will present black, right? The thing will present black, right? If the epistasis gene actually is present, right? What it will do is stop the expression of the gene, right? So it's a possibility that it could stop the expression of the gene itself, right? It could stop the expression of the dominant allele specifically and allow you to appear brown. That is one case there, right? Those are the two cases, right? Or it could affect it in such a way where it kind of creates a in between, right? Something like that, something in between. So what we're saying here now is not that the epistatic gene is dominant, it's just that it controls something else. That is what epistasis is talking about. So the gene itself really does nothing. It does nothing really, but controls something else, right? So I'm not sure if that makes a little bit more sense, right, in that way. So it can mask, meaning it can shut off, right, or it can modify the expression of another gene itself. So in this case with these Labradors, right, one is black, right, black coat large B, one is brown small B, right, so a brown coat, right? But if the gene E is present, right, the gene E is present, what's gonna happen? Gene E determines whether or not the pigment will be black or yellow. So what's happening is that in this Labrador here, in this dog, we have gene E taking over and actually changing or modifying the fur coat from being black or brown, but it actually modifies the expression of the allele to be more yellow. If you get what I'm saying. So the gene is supposed to do something, but the epistatic gene can actually alter what the gene is supposed to be expressing, or it can turn off the gene in the case of albinism. Is it much clearer now? Hopefully it should be much clearer. And that is really what we have there for our genetic terminology. Alright, looking at that. Alright.
It does.